Hello, and a very warm welcome to today's Nucleic Acid Insights webinar, titled Optimizing Plasmid DNA Purification, Strategies for Reliable and Expandable Processes. I'm Charlotte, an editor at BioInsights, and joining me today are Yasmina and Christy, who will delve into the specific technical challenges faced during pDNA production to maximize the yield and purity of the final product while maintaining compliance with regulatory standards. After the presentation, we'll have a live Q&A session, so we invite our audience to pose their questions to Yasmin and Christy using the Ask a Question box at the bottom of your screen, and we'll try to get to that during the session. If you can indicate who your question is for, that's also very helpful for me. So now I'll just briefly introduce our presenters. Yasmina Putz is the Product Manager for Nucleic Acid at Sartorius BIA Separations. She earned her PhD in Biosciences from the University of Ljubljana, specializing in gene identification related to leanness in mice. As a product manager, she continues to enhance production processes for plasmid DNA, messenger RNA, and other nucleic acid-based therapeutics. Christy Franco is the Director of Commercial Operations at VGXI. She holds a bachelor's degree in biomedical engineering from the University of Texas at Austin and a PhD in bioengineering from Rice University. And she has over 8 years of laboratory experience in cell and gene therapy, fermentation process development, and biomaterial design. So now, without further ado, I'm going to hand over to Yasmina to kick off the presentations today.
Thank you for the nice introduction. As mentioned, today's webinar will be focused on challenges that are arising in plasmid manufacturing and on solutions that Sartorius bioseparations can offer to overcome them. In the introduction part, I will outline the importance of plasmid purity and the unique challenges that developers may face when setting up the purification process. Then, we will touch base the importance of having the right analytical tools in order to understand how your purification process is proceeding. Like, do we obtain the highest yields, recoveries, purity, and is our process optimized at the highest level? We will also talk about upstream challenges, touching the lysis and important parameters at this step, and from there we will move to the downstream part of plasmid purification, and we will outline the solutions we develop to overcome challenges appearing in this stage. Okay, so before facing the plasmid purification process and its challenges, let's remember in which applications plasmids are actually used as therapeutics. Plasmids are an essential tool in molecular biology, especially as vectors to deliver genetic material into cells for therapeutic purposes. They may serve as a gene delivery, so meaning they encode therapeutic genes for targeted delivery and expression in cells. Then they serve as a raw material for the manufacturing of viral delivery systems, such as AAV or lentiviruses, and as well they act as a template for the production of mRNA-based vaccines and therapies. So you can see that they are present in three important pillars of the therapeutic applications, and that's why it is crucial to obtain highly pure plasmids. As for example, when you are producing mRNA-based therapeutics, the plasmid serves as a template for mRNA transcription. So here are presented two publications where they showed and explained how, let's call them dirty plasmids, may affect the production of double-stranded RNA, which, when it is introduced in the body or cell culture, can cause a severe immune reaction. In the publication on the left, they outline the presence of short DNA fragments that can participate in the production of small RNA fragments, which align on the target mRNA, and as such, they form double-strand impurities. Same was revealed in the publication on the right, showing how important is to obtain highly pure supercoiled DNA, as damaged pDNA or maybe presence of other pDNA-related impurities, again, can contribute to the production of double-stranded RNA. So, obtaining highly pure supercoiled pDNA to avoid any safety issues when using plasmid for therapeutic purposes is the main goal in plasmid manufacturing, but as such, a lot of challenges can be faced in this process. The figure presents our plasmid purification process workflow, which is designed to enable maximum yields while allowing process developers to quickly implement and optimize it based on their constructs and objectives. This process was created with a clear understanding of the usual problems that occur when setting up purification processes, making sure these issues are handled to allow easier implementation and optimization. Here are listed the important ones, which, when the right solution is implemented, lead to a successful production of highly pure plasmid DNA. For example, developers may face the challenge how to set up the workflow when we want to scale down or scale up, and what brings with it. Then batches must be interreproducible. Methods must be cost and time effective, and of course flexible and generic, so the workflow can be applied on several different plasmid constructs. And additionally, the production must meet the regulatory requirements, as well as the final product must be highly pure, enriched and impurities free. So how can we overcome these challenges and establish an optimized process? So it all begins with the research and development phase. At this stage, the key question is not only how to produce pure plasmid DNA, but also how to do it cost effectively. So in the initial phase of setting up the process, developers integrate their understanding of the construct with various strategies. For instance, using design of experiments in lysis reaction optimization is particularly beneficial for rapidly establishing the optimal conditions for robust, repeatable, and high-yield plasmid production. Then, utilizing a 96 well plate with monoliths can significantly accelerate the optimization process, such as refining binding or elution conditions, or quickly determining the column binding capacity for the established plasmid purification process. Nonetheless, optimization must be backed by robust and reliable analysis to truly understand the process and, of course, to accelerate process development. Another important aspect to consider when developing the process and evaluate the cost of goods is defining the critical quality attributes and key process parameters. This ensures consistent and robust process performance. And once the developed process is optimized, it can be easily scalable. As I mentioned, implementation of plasmid analysis for in-process control helps understanding the process. The PETFIX pDNA analytical platform is designed to characterize plasmids for maximum productivity. Its plug-and-play simplicity allows developers to quickly obtain high-resolution results about their process. A key feature of this platform is its ability to accelerate process development by separating plasmid isoforms and other impurities such as RNA with a single method. And importantly, it enables quantification of the target molecule, allowing you to determine the amount of open circular, supercoiled, or even linear pDNA in your sample. The image on the right displays a pDNA analytical chromatogram of a clarified lysate sample, showcasing the clear separation between RNA impurities and pDNA isoforms. Throughout the process development and scaling up, you can analyze in-process fractions or samples to assess the progress of your process. For instance, you can determine if your lysis conditions are too harsh by observing changes in supercoiled or open circular pDNA levels, monitor the effectiveness of impurity removal during the purification, estimate column binding capacity, and importantly, quantify your final product. On this slide, I have summarized the key points about the PATFIX pDNA platform discussed earlier. The 4 analytical chromatograms illustrate different stages of linear plasmid purification. In the initial lysate material, so in figure 1, you can observe a full spectrum of nucleic acids, including RNA, open circular, supercoiled pDNA, and multimers. As downstream purification progresses, so figure 2, where capture step is presented, RNA impurities are effectively removed by preserving pDNA integrity. In the figure 3, it is presented the sample after the linearization reaction, and neither open circular nor supercoiled pDNA is detected. The PATFIX pDNA platform is also useful to monitor and optimize the kinetics of the linearization reaction, such as adjusting enzyme amounts or reaction time. And finally, the polishing step results presented in figure 4 shows the enrichment of linear plasmid. As highlighted, the PATFIX pDNA platform is really a versatile analytical tool, ideal for optimizing processes and monitoring and refining reactions related to plasmid DNA. Alright, so let's move on to the actual challenges in plasmid production. So here I wanted to highlight the solution we offer for challenges that arise in the upstream, specifically during alkaline lysis. This is the stage where the produced plasmid is released from the cells and is crucial for achieving maximum yield and maintaining the integrity of plasmid DNA. During this phase, the conditions must be really carefully controlled to ensure that the plasmid is efficiently extracted without causing any damage, as inconsistencies can lower yields and compromise plasmid quality, and further affecting downstream applications and overall process efficiency. Developers often face batch-to-batch inconsistencies due to difficulties in controlling factors such as lysis time, precise addition of resuspension buffer or clarification buffer, and mixing. Also, managing large volumes can be challenging. So to address these challenges, we have developed an automated inline lysis system. This system ensures precise and monitor lysis control, reducing the risk of plasmid damage and efficiently removing impurities. Its ease of use allows developers to quickly optimize lysis conditions. And compared to batch lysis, it offers faster process times and it is easily scalable. If the lysis was successful, plasmids are released from cells and most impurities like genomic DNA, RNA, and proteins are removed. However, the plasmid remains in the lysis matrix, which isn't ideal and can lead to degradation. Therefore, further purification is necessary. But also here, challenges may arise, including the trapping and loss of large constructs on the column, the impact of large load volumes and viscous samples on operation time and recovery, and the difficulty in separating structurally similar impurities like RNA or genomic DNA, which can bind to the column and co-elute with the target. Furthermore, enriching the target supercoiled pDNA by removing other plasmid isoforms presents additional difficulties. Thin chromatography monoliths, such as DEA or C4HLD, are excellent tools to address these challenges. They provide high resolution in removing unwanted plasmid isoforms, offer high recoveries, can process large volumes, and have short process time due to fast flow rates with low break pressure. The columns are also stable in cleaning buffers, allowing their reuse or recycling. They are also easily scalable from experimental or process development scales, like 1 ml column volumes, to manufacturing scales where 8-liter columns can be utilized. So the DEA column is a weak anion exchanger, and in plasmid process is used as a capture step where RNA, proteins, and endotoxins are separated from the plasmid, and at the same time, high purification efficiency and high recovery rates can be achieved. The plasmid can be bound on the column by buffer exchanging the lysate to binding buffer, or a more straightforward and simpler inline dilution approach can be applied to reduce the processing time and to decrease process steps. The DEA column is available in 2 channel sizes. The column with 2 micrometer channels is suitable for plasmid up to 8 kilobase pairs, and for processing lower volumes, like when buffer exchange and concentration of clarified lysate was applied, and as well as high binding capacity can be achieved with these channels. If the sample is more viscous or contains larger plasmids or sensitive constructs that need faster processing time, then the column with 6 micrometer channel size would be more appropriate. So here are presented 2 chromatograms where plasmid capture was performed on DEA with 2 micrometer and 6 micrometer channel size. In both cases, more than 90% pDNA recovery was obtained in only 3 column volumes. Endotoxins, proteins, and RNA were successfully reduced or removed. And as for the DEA with 6 micrometer channels, a flow rate more than 3 column volume per minute was applied, with negligible increase in back pressure that allowed a load of more than 100 liters to be processed in a really short time. As mentioned before, also the analysis using PETFIX pDNA platform confirmed the complete removal of RNA using both types of DEA columns in flow-through fractions that are presented in blue and green line. And in eluate, pure plasmid with high integrity was obtained, as you may see the sample in the red line, where there is no presence of RNA impurities. So the captured supercoiled plasmid or linear plasmid must then be further separated from the other isoforms. This is obtained with the polishing step on a hydrophobic simultus C4HLD column. This column allows separation and enrichment of supercoiled and linear pDNA from the remaining impurities, as you may see on the figure on the right. Two strategies can be applied using SIM C4HLD column for pDNA polishing step. The first is the bind-elute chromatography, where all isoforms, so open circular and supercoiled pDNA, bind on column at high concentrations of ammonium sulfate, and when decreasing the salt concentration, then different isoforms elute. The second strategy, which was published and nicely described in the paper listed at the bottom, uses selective binding approach, where by adjusting the binding conditions, only supercoiled plasmid binds on the column, while other isoforms as well as impurities don't. The method is also more beneficial as lower salt concentrations are needed, meaning also the cost for chemicals are reduced. The method is scalable, GMP compatible, and 100% homogeneity of supercoiled pDNA is achieved without having traces of other isoforms. Let's look on a case example of the mentioned SHIC method. 400 ml simultus C4HLD column was used. Sample was loaded at 1.5 molar ammonium sulfate, and the enrichment of supercoiled pDNA was obtained in step gradient at 0.9 molar ammonium sulfate in only 3 column volumes with 91% recovery. The successful removal of isoforms was also confirmed by using the PET-fix pDNA. So here is presented polishing of 7.5 kilobase per plasmid. In the upper panel of figures, so A and B, is the preparative chromatogram of loading part in figure A, and in figure B is the elution of supercoiled plasmid. The bottom figure is presenting the analysis with PET-fix pDNA. The red line is the load, so the DEA elution, and you may see the presence of both, so open circular and supercoiled isoforms. This sample was then loaded on the C4HLD column at a certain concentration of ammonium sulfate. And in the flow-through fractions, so the black and blue line, you can see the presence of open circular isoform, meaning it did not bind on the column while the supercoiled did, and it is observed in the elution fraction presented in the green line. As you can see, this approach not only reduces the amount of ammonium sulfate used, but also results in the recovery of enriched and purer supercoiled pDNA. It is also important to highlight the overall effect of purification using simultus monoliths. The discussed process achieves over 80% yield with approximately 90% homogeneity of supercoiled plasmid in both capture and polishing steps. Impurities are reduced to low levels or even below detection limits, as illustrated in the table. So, in summary, the purification process using the simultus DEA column for capture and the C4HLD for plasmid polishing produces highly pure pDNA ready for use. As showed in purification and polishing examples, this developed process enhances productivity and reduces manufacturing costs, and offers a straightforward and scalable solution for the separation of all plasmid sizes. In conclusion, overcoming challenges in plasmid manufacturing requires a combination of automation, innovative approaches, and supportive expertise. This leads to rapid development of in-house expertise and innovative solutions tailored to pDNA applications, which can eliminate difficulties and enhance next-generation processes. Additionally, it is crucial to develop the process with clearly stated goals to ensure successful implementation on a larger scale.
Thank you for the introductions. As Director of Commercial Operations, I am excited to walk through in this part of the webinar how VGXI has implemented new technologies in our workflows to adapt to the challenges that we've seen with plasmids for next-generation therapeutics. Valued partners like Sartorius are a critical factor in driving these innovations forward. These slides will cover an overview of our service portfolio and expertise, how plasmid DNA plays a role as a critical starting material in next-generation therapies, the strategies that we've implemented in order to optimize our downstream purification process, and in particular a case study in how this has been applied to adapt the Sartorius monolith technology in our workflow. Finally, we'll summarize key takeaways. VGXI was established in the US over 20 years ago with a historical focus on the GMP production of DNA vaccines, so plasmid more as a bulk drug substance and supporting in-house GMP fill finish for DNA direct injectables. Over those years, we have developed extensive expertise as a pure-play plasmid-focused CDMO, supporting our clients in clinical trials around the world. We have two facilities. Our flagship site is north of Houston in the Woodland, Texas, and our new headquarters and Phase 3 commercial production site is located about 20 miles north at Dyson Technology Park in Conroe, Texas. The key values that we provide to all of our clients is in our customer-centric approach and the track record we have in pursuing manufacturing excellence. This is supported with leading-edge commercial capabilities, particularly incorporated at our Conroe site. That includes state-of-the-art equipment and automation to make sure those programs can be supported all the way through commercial supply. Despite our growth, we are still an organization that understands we need to keep the customer in mind at every step. That means being able to be agile and flexible and really adapt to those program needs and timelines. We know that it can be critical for those timelines to the success of the clinical program. Finally, VGXI has the scale necessary to make sure that when those programs reach the later development stages, we can support them every step of the way through 1,500-liter large-scale manufacturing. So I'm sure most of the audience today is well aware of what a plasmid is, but I'll take one quick slide to step back on what it is that we're manufacturing. So really it boils down to small, circular, double-stranded pieces of DNA that are supercoiled and amplified within E. coli bacterial culture. Plasmids are an essential starting material for a wide range of next-generation therapies, and that includes AAV or adeno-associated viral vectors, lentiviral vectors, as well as non-viral vectors for gene therapy. They're also used to gene-modify cell therapies like CAR-T, and they are a critical starting material in mRNA manufacturing as the linear template. Finally, back to our historical experience, the plasmids can be the direct API themselves, and they're used in vaccines and immunotherapies. When I joined VGXI 10 years ago in 2014, our focus was really in those latter categories of immunotherapies and vaccines. And I'm presenting the data here to show really what was happening globally in terms of the increasing development of cell and gene therapies. This chart shows the rise of the clinical pipelines, primarily at that preclinical phase, but also maturing into those later phase 2-3 programs, and of course the first commercial approval of a gene therapy with Kymriah in 2017. So VGXI saw this reflected in the types of manufacturing projects that were being requested by our clients and supported at our facilities. And we've really developed a robust service portfolio to provide those phase-appropriate services across development stages, including through to marketed therapies. These core services start with our preclinical plasmid manufacturing, which includes anywhere from a 10-liter or less up to about a 100-liter manufacturing scale to supply up to gram quantities of high-quality preclinical grade for those early studies. We have an intermediate grade, which is highly documented, or HD production, and that supplies GMP-like plasmid, typically used by our clients who are requesting it for critical raw material applications like AAV. And finally, the full GMP manufacturing services covers the range from 10-liter all the way to our commercial 1,500-liter fermenter, so large-scale batches for commercial supply. Over the years, we have produced over 325 batches for human clinical use, and that equates to over 3 kilograms of plasmid DNA, not counting those HD and preclinical batches. A key aspect for every one of these core services is that we use an equivalent or the same manufacturing process. We use the same raw materials. We test across the same full characterization panel, and we expect that same specification profile on the final product. So when our clients come to us and order preclinical services, they know that the material they'll be testing in their early preclinical studies will be representative of the GMP grade material that they'll use in their later stages. I showed on the earlier slide the shifting global trends in cell and gene therapy clinical development, and we saw this within VGXI reflected in shifting production trends and shifting requests from our clients. So in 2014, where close to 80% of the product lots manufactured were for direct injectable applications, that portion has adjusted to over half of the products manufactured today are critical starting material, in particular for gene therapy applications. And these come with considerations of larger payload sizes, typically larger plasmid sizes, as well as other complex regions, secondary structures, GC-rich, or the inverted terminal repeats that you would see on an AAV gene of interest. Understanding where these challenges are and getting in front of them with how you approach a program can be critical to making sure you're successful. VGXI's manufacturing process starts before any of the steps in this slide with transformation, careful colony screening, and selection, and then establishing a well-characterized master cell bank. The cell bank is then brought into the actual production process at the upstream step, with anywhere from 10-liter up to 1,500-liter scale fermentation. Coming out of that fermentation, a very critical step is cell lysis, and this is where VGXI's patented AirMex technology is incorporated to allow for gentle but very efficient separation of the plasmid from host cell debris. It's a continuous process with lysis, neutralization, and primary capture that can be scaled all the way to that 1,500-liter with the same outcome and product. So it's very controlled in terms of the exposure to those lysis conditions and the efficiency of mixing and neutralization to achieve consistency across those scales. Downstream purification then includes chromatography steps to remove those impurities, including the host cell genomic, DNA, RNA, protein, and endotoxin, with a final concentration using tangential flow filtration, or UFDF. Focusing in on lysis, neutralization, and primary capture, which is the first part of the downstream process, the anion exchange is a critical piece and allows for capture of the plasmid DNA and wash-through and removal of a large portion of host cell impurities. Historically, VGXI has used a membrane-based approach which can provide very reliable product quality across a range of plasmids. And typically we were working with immunotherapy or vaccine plasmids, which were high yield, high copy origin, and typically under 10 kb in size. But as you saw on the market trend slide and our own internal manufacturing trends, we were beginning to work more and more with challenging plasmids that were larger, with more complex sequences, and we saw an opportunity here to optimize the process at this step based on occasional reduced or inconsistencies in the yield output across the anion exchange. This was particularly for those larger sequences, or in some cases, if we were working with a high culture density, such as fed batch process, we would see that reduced yield. And root cause was narrowed down to potential restriction of the material flowing through based on the size of the pores within the membrane. So for those anion exchanges, it was a less than a 1 micron pore size, as well as possible irreversible binding or membrane fouling, which can lead to increased back pressure, and in some cases, dramatic yield loss as a result. We identified this very early on as an important opportunity to innovate in the downstream process and optimize the best support plasmids for those next generation therapies. We reached out and worked with key partners and collaborators, and in particular, Sartorius and BIA, based on their focus on innovation and excellence in their services and product portfolio. It was recognized within that portfolio that the DEAE monolith could be an ideal technology suitable as an alternate primary capture modality. The reasons for that really include the range of pore sizes that are available with the monolith. So it's a solid sponge-like matrix with controlled average pore size anywhere from a 1.3 micron up to 6 micron. And with working with those larger plasmids, you have that ability to shift to the larger micron pore size and reduce possibility of DNA being restricted during processing. The bed volumes also come in a range of scales, from a 1 ml development for small testing up to 8 liter or even 40 liter for commercial supply needs. And related to those commercial supply considerations, the column can support cleaning and reuse, for example, in the case of a resupply run. During the optimization and testing, we looked at a wide range of different factors, but to focus on some of those key points, include the effects of various modeled load conditions, impact of different wash conditions, the yield considerations if we reach that saturation point, and aspects to reduce the kind of upfront RNA load, so you can focus those binding capacity on the target plasmid DNA versus total nucleic acid. For these next couple slides, I'd like to pull out and show some example data collected during this evaluation. And we really looked at a broad range of different plasmid types, anywhere from smaller 5 kb plasmid up to nearly 19 kb construct, covering a variety of different AAV helper, RepCap, and gene of interest plasmids, as well as some non-AAV plasmids to really rigorously test how the technology performed. An example of the data is included in the right in terms of yield analysis between the 5 kb and the 13 kb pDNA, where we're using the applicable 2 micron pore size for that smaller plasmid and the larger 6 micron pore size for the 13 kb sequence. For larger DNA sizes, it's expected to see some reduced yield, but we saw as much as 70% overall purification yield with that 5 kb sequence, and an overall 30% yield for the 13 kb sequence. And I'd like to emphasize here, this was some of the early data. There's been optimization since to avoid reaching column saturation during loading. When that saturation point is reached, it can reduce that yield due to a portion of your target plasmid DNA flowing through with those wash or flow through fractions. Staying under that saturation point helps ensure that all your DNA that's loaded is captured and retained all the way through to elution. Comparing back to historical purification yields for some of these very large sequences when they're over 12 kb or more, we've seen as low as 10% or less. So observing 30% to 70% yield is a significant improvement over some of the alternate technologies that we've used previously. We did complete full characterization panels on the final purified product to make sure that it was comparable to the specification profile observed using the membrane-based modality. And whether it was a 2 micron or a 6 micron and across different plasmid types, we saw that the specifications were met with the target impurity profile and high supercoil. Taking the same 2 example plasmids from the prior slide, we also looked at the ability to clean and reuse the monolith across runs. This is important if you want to use it for resupply later, but also more critically within a production run. If you have a large quantity of cell paste from a fermentation, you can run multiple loads within the process and achieve consistency in the binding capacity across those loads. It was tested up to 6 runs, and for both the 2 micron and 6 micron monolith, the average binding capacity matched those expectations based on the Sartorius manufacturer's information. And you can see there is that expected lower binding capacity when you're working with that larger pore size. Then next over on the mass balance recovery, this was a check to ensure that there was no irreversible binding or column fouling occurring during the repeated runs. We could look at the total nucleic acid quantity that was in the load fraction prior to start, and then identify that we were recovering over 96% of this material by evaluating that flow-through, wash, elution, and regeneration fractions post-run. The elution fraction comprised the majority of this material as plasmid DNA. So overall, the data shows and supports the ability to consistently clean and reuse the monolith, either within that production run for processing of larger cell-based quantities, and also you can regenerate, clean, and reuse for future resupply runs to reduce the COGS considerations when you're implementing this technology. In summary, VGXI has established and implemented this monolith technology in our downstream process to provide additional flexibility and options, especially for larger, more complex sequences. It's shown to increase overall yield as well as batch-to-batch consistency, especially for larger plasmids. And for high cell culture densities, such as a fed batch approach, it can reduce that risk for column fouling. We have shown with data that multiple loads can be run on the same monolith so they can support full processing at scale. And Sartorius offers a 40-liter version of this technology, which is particularly well adapted to our 1,500-liter fermentation. When implementing the monolith, there is consideration of the increased upfront material cost. We've shown that the ability to clean and reuse a column can balance this out. For example, the column can be reused from an engineering GMP run or for future resupplies when you're making the same product. Finally, for the analytical profile, it met all the same release criteria and expectations compared to the prior technology. And some of the early data showed there was slightly higher RNA, so we've continued to optimize and evaluate potential for RNA reduction, which can also better apply the column binding capacity towards our target plasmid. Thanks everyone for joining in this webinar today. VGXI as an organization is driven by innovation. We place a premium on quality for our clients, and we leverage best-in-class technologies. We work with partners who share in those same values like Sartorius. Their monolith chromatography technology provided that innovative tool that was necessary for us to deliver our promise to our clients. And the partnership that we have with Sartorius and others in our industry ensure that VGXI remains at the forefront of excellence and reliability. I'd like to call out here when you're choosing your partner, whether you're going to be working with VGXI in your manufacturing or some other partnership, make sure that they have the right technical expertise. It is going to be critical to understanding and predicting what those challenges are so you can get in front of them to mitigate potential risks. Consider also your long-term goals for the clinical program and make sure it matches with the scalability for your partner's capabilities. Finally, always take that collaborative approach. That's what's going to be driving the success and ultimately bringing your product to patients in the clinic. Thank you everybody for joining today, and thank you in particular to our partner Sartorius in supporting this webinar. I have a contact code here if you want to come to our website and learn more about our services, or you can reach out at info@vgxii.com. At this time, I'd like to open it up for any questions. Thank you.
Thank you very much, Christy and Yasmina, for great presentations. Before we start our Q&A, we'd just like to ask the audience a quick question, which should be popping up on your screen around now. So I'll give you just a few seconds to answer that before we start the Q&A. Okay, brilliant. Thank you very much for your input. Now we're going to start the Q&A. So I would invite Yasmina and Christy to join me on camera. Hopefully you can see us. But at any rate, let's unmute and start the Q&A. So, okay. So first question I've got here is for Yasmina. What are the best practices for removing RNA and genomic DNA contaminants?
So the best practice would actually be the development of a robust upstream part, so like the lysis part, because if this part is developed well, you can already reduce the majority of impurities, and by doing this, you also facilitate
the downstream process, so the purification process, because you load, so you bind less impurities on the columns, which as well actually improve the all over purification performance then.
Perfect, thank you, Yasmina. So next question I've got here is for Christy. How do the different anion exchange capture formats compare when it comes to cleaning and reusability and robustness?
Great, yes. So we have a lot of historical experience working with the membrane-based format for that primary capture. And while there are kind of those cleaning and reuse protocols that come with that format, in practice, what we've seen with a range of different plasmids is there's not as much consistency in being able to fully clean and get back to the full starting binding capacity. So for that reason, we've really used that technology as single use across whether we're doing preclinical runs through large-scale GMP runs. For the monolith, what we saw in the slides and the data is we can perform the bind dilute cycle, so we can process large volumes by doing multiple bind dilutes. We can also do the full cleaning and regeneration to bring that back to the starting binding capacity. For example, if we exceed that saturation point. So it really has proven to be a more robust technology for that reuse purpose.
Perfect, thank you. Yasmina, back to you. How can I scale up the plasmid purification process without compromising quality?
Maybe as I mentioned or highlighted in the presentation, it is important the development stage. So the experiments when defining the process development, that experiments are well defined, and maybe keep in mind the scaling up risks, and perhaps well defining the quality attributes. And of course, another important thing is to implement a robust analytical method.
Brilliant, thank you. And Christy, why is determining column saturation point critical to achieving good yields, and how is that evaluated during the development work?
Yes, thank you. So that's a really critical and important piece of the process development before we move into the clinical production runs, is identifying what that saturation point is. It varies for each plasmid. Each different sequence behaves slightly differently on the column, so you want to know what it looks like. If you are not reaching the saturation, then you're going to not fully utilize the binding capacity of the column itself. So you could have to do additional binding dilutes or less efficiency of the runs. If you end up exceeding that saturation point, you are going to have the desired product coming out in either the flow through or the wash step. So your goal is to get up to that saturation point to really maximize the efficiency of the column as you're using it in the production.
Absolutely. Okay, Yasmina, how do I select the right channel size of DEAE column for plasmid capture?
So usually we recommend to use the 2 micrometers channel sizes for plasmids up to 8 kilobase pair. But also it depends on the conformation of the isoforms, or maybe the viscosity of the sample and the stability of the sample after lysis. And in this case, we suggest to use the column with larger channels.
Brilliant. And Christy, for programs moving into larger scale clinical or commercial production, what are some of those main scale-up considerations?
Absolutely. So really, it's key to kind of keep the end in mind and understand what you're going to be scaling to in the future, so that from the very beginning, you can be planning that with what you're implementing for your unit operations, who you choose for your partner, and their capabilities, and getting you to that commercial stage, as well as investing kind of the time and effort in that early stage development and optimization of the process itself. So taking time before you start even your first clinical batches to optimize the upstream yield, your specific yield of the fermentation, that will get you the best efficiency as you move into those larger fermentation scales and getting the amount of product out for your commercial program.
Perfect. And Yasmina, how do I optimize the lysis conditions for maximum plasmid recovery?
Yeah, so maybe optimizing each parameter in the lysis procedure. So time of lysis, concentration of neutralization buffer, then maybe concentration of precipitation buffer. So this each parameter should be optimized, and yeah, again, it is important again to really have a good method, a robust and reliable analytical method, because only with this part you can then optimize well the whole process. Otherwise, it can maybe mislead you to the wrong conclusions.
Interesting. And we've got time for one more question. Christy, in working with larger or complex plasmids, what are the key challenges? What's the best way to de-risk the program from the start?
Absolutely. So, one of the key things is making sure you're working with a partner that has experience with those kind of similar sequences. AAV plasmids, in particular, the ITRs, can pose challenges that if you haven't worked with them in the past, maybe not have the right tools to look for the integrity of the ITR regions during the development of your manufacturing and your purification process. Again, kind of like with planning for scale, you want to take that time and invest in the early development and process optimization. So looking at your upstream process, evaluating different cell lines, making sure they're compatible and you're not getting deletions or changes in your sequence, especially larger plasmids have more tendency to go through recombination events. Evaluating those fermentation conditions and optimizing for the specific yield during fermentation. And then, that downstream purification approach, just like Yasmina mentioned, your columns and your chromatography setup should be appropriately matched to the size of the plasmid to make sure you're not going to have restricted flow or yield loss just because of the size and restriction during those steps. So there's a lot of different aspects of it, both upstream and downstream, but investing the time early to look at those critical stages will help you be successful when you go then to take that process to the larger scales, because you don't want to be rushing to get to the end, trying to brute force something never works out well. So invest in that early development stage.
Absolutely. I think everyone would agree with that. This is a good note to end on. So thank you very much, Christy and Yasmina, for answering those questions. We're going to wrap up there for today. Any questions that we didn't get to, we'll reply later by email. The webinar will be available on demand from tomorrow, so look out for an email from us with the link. We'd also like to invite you to take a short survey to provide us with some feedback on today's webinar. The link to that can be found in the resources on the right-hand side of the screen, or it's also going to be in the on-demand email, so look out for that. There's also an attendance certificate available, and that's also just on the right-hand side of the screen there. So all that's left then is to thank Yasmina and Christy once more for a great presentation, and thank you very much to the audience for watching. We do hope you'll join us again soon.
Great, thank you.